Okay, so welcome to this introduction to bladder cancer. So this is uh, aimed at supporting students starting in my group, um, some of the aims of our research, and some of the content that you'll need to understand as your project gets started. So firstly, a bit of anatomy. So our urinary system has two distinct zones. So we have the upper urinary tract, which is the kidneys and the ureters. They're the pipes that connect the kidneys to the bladder. And then our lower urinary tract, which is the bladder going down to the urethra. So this system obviously has a lot of work to do. It helps to protect our bodies against pathogens. It has to handle all the toxins that the kidneys have filtered out of our blood and into the urine. And then during the course of making the urine, moving the urine, and storing the urine, it has to accommodate a lot of changes in pressure and size and shape. Now, the tissue that we really care about when we're thinking about the cancers that arise within the urinary tract is the urothelium. So the urothelium is a highly specialized epithelial layer that lines the whole urinary tract from the inside of the kidneys all the way down to the top of the urethra. Now, there's an H&E image here showing different layers of the urothelium. Now, in this case, the lumen, so where the urine would be, is at the top. That's typically how we orient any of our histology slices. So we've got different cell types here. We've got superficial cells at the top. They're sometimes called umbrella cells. You can see that there is an apical surface here that helps to protect, forms a really tight barrier against the urinary toxins. Sometimes these cells have got two nuclei, so you can see that on the left-hand side here. We then have at the bottom what these cells are called basal cells. So these are the ones which are touching the basement membrane. So they're called basal because they're at the bottom of the tissue, not because they are more primitive. They're specialized for a different job. In between the basal cells and the superficial cells, we have these intermediate cells. And depending on how full the bladder is, how stretched the bladder is, the thickness, the number of cells that appear in this layer can range from between 1 to 5 to 6 cells. Now, underneath the basement membrane, that's where we have our lamina propria, where the stroma is. So we have our blood cells, our nerve cells, fibroblasts, those kinds of things are all there supporting the urothelium. And then underneath the lamina propria, that's where we get our layers of muscle. You can see some of these darker dots in the plot here, and these are immune cells, which have come from the blood vessels and come into the urothelium. These have a role in maintaining the tissue and in innate immunity, but they are separate from the epithelial cells, and you can see that often by the size and the shape. So one thing to note about urothelium is that it's quiescent. That means that it's not really an active cell cycle. It's very long-lived. Cells will live for around 100 days. Um, and only 1% of the cells are in cell cycle at any given time. You will read papers, and it's fine to read these papers and think about them and critique them, where where people will give evidence for a urothelial stem cell. And they also present this idea that basal cells are more primitive, they form a kind of stem progenitor population, and that as you go up the urothelium to the superficial cells, this is a kind of terminal differentiation. Now, the work that we've done in the Jack Birch unit here at York for many years has never really shown evidence that there are distinct populations of stem cells. In fact, our work would show that any individual cell within the urothelium is able to recapitulate the whole layer, and it's just a case of where the cells are that determine the protein markers that they have, and so the specialization that they have. So our cells on the top, our superficial cells, they are more specialized for blocking urine, and our basal cells are more specialized for adhering to the basement membrane. It doesn't necessarily suggest a trajectory. The other thing to be really careful with here is that we will talk about cancers, and papers will talk about bladder cancers as being basal or luminal, talking about some of the differentiation markers. Basal in the context of cancer means primitive, not at the bottom. A basal cancer does not necessarily come from the basal cells of the urothelium in just the same way as a luminal cancer does not come necessarily from the luminal cells of the urothelium. It's a bit of unhelpful mixed language that's used in the field. So I've said bladder cancer and I've said urothelial cancer. Now, the important thing here is that because the urothelium stretches from the renal pelvis of the kidney all the way down into the bladder, you can have urothelial cancers which are not found in the bladder. Now, the rates are quite different. 92% of urothelial carcinoma are found in the bladder and only about 8% in the upper tract. Um, there are differences in terms of how they present. So our upper tract cancers are much rarer and they mostly present at a muscle invasive stage. This is a grading system here at the bottom that we use. Um, the bladder cancers, however, most of these, so 80% of them present at non-muscle invasive stage. Now, these can be treated quite readily, either with intravesical chemotherapy or by surgical removal, but they recur, so they come back or new tumors form depending on who you speak to. And then some of these tumors will go on to become muscle invasive. So what actually causes urothelial carcinoma? So remember that the urothelium is bathed in the toxins of the urine, and so the urothelium itself has an incredibly high mutational burden, so it has lots and lots of mutations. So what is causing these mutations? Well, Cancer Research UK would say that a lot of mutations are caused by smoking. And it's certainly true that smokers do get bladder cancer at rates higher than non-smokers. But the relationship isn't quite perfect. So if you were to look at lung cancer, for example, here on the right-hand side, the more that you smoke, the higher your risk of getting lung cancer. With bladder, that's not quite the relationship. So with bladder, the more that you smoke to a point, that is where you increase your risk of bladder cancer. But if you keep on smoking and increase the numbers of cigarettes you smoke, your bladder cancer risk doesn't actually go up. So the relationship isn't quite there. Some of the other common risk factors that we have at age, so smoking, bladder cancer is a disease of aging, but also it's a disease that's found predominantly in men at a 3-to-1 rate. Now, traditionally, this was associated with differences in smoking, but also exposure to different chemicals in the workplace. But as people's working patterns have changed, some of these percentages aren't quite right. So that's led us to want to look to try and find other reasons for what is causing bladder cancer. One thing to remember when you're reading the literature is that this talks about bladder cancer in Europe and North America predominantly. If you were to look at a paper on bladder cancer from Egypt, for example, most cases of bladder cancers are caused by schistosome worms. So it's a very, very different disease trajectory. So one way that we can try and work out what's causing a cancer is by looking at the types of mutations. So different types of mutations, so changing from a C to an A or C to a G, for example, they're caused by different mutational processes. So for example, C to A changes are commonly associated with smoking, and C to T changes are commonly associated just with aging. Now, we can look at the mutations like this, but another feature that really changes how these mutational patterns form are the bases either side of the mutated base. So the base that precedes, so the base to the 5' or the base to the 3'. And this is all about what the DNA structure looks like to different chemicals that could come in and mutate the DNA. So that means that there's 6 different mutation types. So C to G, C to T, C to A, etc. And then there's different bases that could be either side, giving 96 different mutation classes. Now this makes a very big bar graph. The key thing here is to look at shape and the colors. So if we look in lung cancer, we can see our C to A changes predominantly associated with smoking. This is very characteristic of lung cancer. But with a bladder cancer, we don't see that pattern in the blue region at all. You can see in bladder cancer, it's very flat. What we do see in bladder cancer are these very kind of characteristic bars in the black region and the red region. So these are C to T predominantly and C to G changes. And if you were to zoom in a bit and focus in on these patterns, it's C to G when it's preceded by a T, or C to T when it's preceded by a T. So this is the chemistry. Now, these kinds of mutations have been associated with a series of enzymes called ApoBec enzymes. Now, ApoBec enzymes are antiviral enzymes. So this mutational pattern, this mutational signature, is one that you'd commonly associate with cancers such as cervical cancer, which are associated with HPV. But bladder cancer doesn't have a viral cause as far as we were aware before this mutational work started happening. It's associated with smoking. So how do we get this mutational pattern but no virus? Well, that has been the work of one of my colleagues here at York, Simon Baker, who's been working to try and find the viral cause of bladder cancer. I mean, the exciting thing about that obviously would be that if we can find a viral cause, we might be able to vaccinate and prevent a lot of these viruses. So he's been working on this virus called BK polyomavirus, and he's described it as a hit and run driver for bladder cancer. So the idea here is that the virus is dormant in the kidneys. Every so often during life, it reactivates, causes damage in the urothelium without itself getting stuck in those cells. And over a long period of life, those bits of damage accumulate over time, and eventually you would get a cancer. The idea then with smoking is that the immune system might be dampened, and so the virus might be released more often in smokers than in non-smokers. And it might be that then the different kinds of smoke carcinogens in the urine get handled quite well by the urothelium, but effectively it's a distraction allowing the virus to cause more damage. Now, it's really, really important for our understanding. We typically you'll find lots of cancer papers where we're looking at mice as a useful model. But for bladder cancer, mice aren't the best model. So we need to really focus on human data that's really relevant to us. So particularly for BK polyomavirus and the ApoBec enzymes that cause bladder cancer, mice don't have the relevant ApoBec enzymes. They don't get a virus that's analogous at all. So the models that we have in mice of bladder cancer are all induced by chemicals which create really aggressive cancers that develop very, very quickly, which isn't matching the disease progression at all. And actually there's quite a few differences in the urothelium and the bladder function of rodents. So regardless of exactly what's causing bladder cancer, the key feature here is that bladder cancer gets a lot of mutations. So bladder, you can see on the right-hand side of this plot in terms of how many mutations per megabase of genome, gets a lot of mutations, second only to lung cancer and melanoma. So melanoma, lots and lots of damage from UV, and then lung cancer, lots and lots of damage from smoke carcinogens. Now, it's not only that we get lots and lots of mutations, but it means that the genes that get mutated and the genes that drive the cancer can be very different as well. This makes it really difficult to understand which pathways we should try and target with drugs. We don't know how different mutations interact in different tumors. It makes it quite difficult to work with. So bladder is actually one of the most diverse. For comparison, I've got ovary here and pancreas, which have some driving mutations found in many more of the cancers, but it allows you to focus on one thing. So P53 here, known as the guardian of the genome, is a really important gene mutated in lots and lots of cancers, and there are drugs available to some of the mutations in this gene. The problem we have with bladder is that there are so many different ones that we could work with. So why do we need to group people at all? Well, it's all about personalizing treatment. We need to understand which tumors will recur, which early tumors will progress to the aggressive muscle-invasive disease, which aggressive muscle-invasive cancers will then metastasize and go on and go to different parts of the body. And then across all stages, how we actually pick the best treatment for the best patient. Now, in the projects that we'll be working on at York, we're going to be focusing on muscle-invasive disease, mostly to do with treatment selection, but also trying to understand metastasis. And the major dataset we'll be using is the bladder cancer dataset from the Cancer Genome Atlas, which is all muscle-invasive cancers. So in order to understand treatment selection, progression, metastasis, It's all about grouping patients into clinically relevant groups that we can target in the clinic. That's the main aim of our research, not to just be academic, but to actually go into the clinic. So stratification is the problem. How do we put patients into groups? So the reason for wanting to stratify is that if you have a single treatment of one fits all, you will have some people who respond very well to a drug. So there's good efficacy here. Some people won't respond at all, and some will have really, really bad side effects to the drug. Now, we often look at the bad side effects as being the major negative here, which is absolutely right. But remember that the people who don't respond at all, their treatment has then been delayed for 6 months. And in a group of people where 50% of them will have died within 5 years, it's really important to get the treatment right the first time. So the idea with stratification using sequencing data is if we use the right data, we can find groupings and we can personalize treatment either to give different treatments or to make sure that people get a treatment that's going to work for them first time. Now, the way that we do this in a computational sense is with unsupervised clustering. This is a machine learning technique that's used to find order in samples where we don't actually know the answer. We don't know the groups that we're trying to find. So people have tried to use mutations to do this. The problem with mutations in bladder cancer is because there's so many, and because there are so many different types of mutations, there aren't nice clear groups. A group of patients that are mutated in TP53 but nothing else. You see that there's overlap with each column here is a different patient. And there's lots of overlap between different mutations, and then you'll have groups of patients where there are no mutations in these common genes. So it's quite difficult to know what the impact of each mutation would be and how different mutations would interact. And then most of the mutations we find in cancer genomes are only found in 1 or 2 patients. So it's very difficult to personalize. At the moment, we can't really personalize to every single individual. We still need to find groups that would treat lots of people. That's just the economics of scale for finding new patient groups. So gene expression data, like RNA sequencing data, was used to try and understand these cancers. And this happened a lot in cancers in the 2010s. And it happened a lot with bladder cancer. It wasn't other cancers, it was bladder cancer as well. So there was this explosion of papers in the sort of mid-2010s, all proposing different strategies for classifying tumors. Now, whilst the groupings they came up with were different, the same approach was used. So you take a piece of tumor and you do some gene expression profiling like RNA-seq. The problem here is, have you only sequenced tumor or do you manage to get surrounding tissue as well? Is there surgical sampling or is it actual biology? Are there immune cells in the tumor or are they in the neighboring tissue? You normalize your data, you get rid of genes which have low expression. You then select genes which have really high variance. They vary a lot across your cohort, so they have a lot of power to be able to separate samples. That's fine, but if you have variability based on cell types that aren't just tumor in there, you can create entire groups. So this stroma-rich group that you'll see in the literature is effectively not really a proper group, but just a group that had more immune cells or stromal cells in it. And then you can try and use these genes with high variance to find subgroups, and then you try and hopefully link these to markers in the clinic, but that is often very, very difficult to do. Now, to get past all of these issues that we had with multiple different classification systems, in 2020 there was a consensus strategy brought together. And you can see in this plot on the left, the different colors represent different strategies for grouping tumors together. And essentially, shared features we use to bring these labels together to make a single consensus classifier. And that's the colors that you'll see on the right-hand side. Now, this is used a lot now in the literature, but it hasn't, whilst it's called a consensus, groups have continued to try and develop strategies beyond this because this one has a few problems, particularly for trying to link to the clinic. So again, mutations still very divided between these different subgroups. There were some sort of biases. So for example, FGFR3 mutations are predominantly found in this luminal papillary group, this sort of light green group. And TP53 and RB1 mutations are typically found in this basal group, but it's not perfect. There are some differences in survival. So these purple neuroendocrine, but there's only 6 of these in the cohort. These samples do particularly badly. Basal tumors do particularly badly, and our luminal papillary tumors do the best. But the separation between these lines isn't very big. There are some suggestions that basal tumors, for example, respond better to immunotherapies than luminal tumors do. So there have been some trials suggested based on different classification approaches. But we're yet to see, because of the fact that these groups are clearly not really that clinically relevant, we're yet to really see if these are going to help people in the actual clinic. The problem still remains that these classification systems, as complicated as they become, they're effectively measuring 2 things. How differentiated our cancers are. So are they primitive? Are they basal, quite primitive? Or are they more luminal? Do they still look like the original urothelial tissue? And then how many immune cells do they have? So this classification that I've made on the left-hand side here, the x-axis is differentiation. How differentiated are they? And the y-axis is how many immune cells. And I hope that you can see that when I've overlaid it with the colors from the consensus classification, effectively, your luminal tumors are all on the right-hand side with high differentiation and low immune status, and your basal tumors are all on the left-hand side with low differentiation status and a bit of immune difference. So if we can match the consensus classification with just 3 genes, it really isn't working for us. So machine learning is giving us very little additional information compared to these markers, these gene markers that we've known about for decades. The subgroups are arbitrary. We get approaches like to go. And when you change that you use when you're matching genes to particular tumors that are at the edge, so the ones sort of down here, the green and yellow ones here, depending on which annotation you use, they would be a different subgroup, which means in the clinic, they would get a different treatment strategy. The problem is that these subgroups are academic, they're not clinically focused. So essentially, what can we do to fix this? The big problem as I see it is that we can't rely on data alone when we've got so much biological knowledge already. That biological knowledge is essential to direct our questions. And what I've done in my lab to try and fix this is by bringing the biology in. So the standard approach for machine learning for subtyping is what I've shown already, this in the gray. On the right-hand side here, we have this biology-driven approach. This is what my PhD student Vlad did in his project. So what he did is, instead of using tumor for the expression profiling, we used healthy tissue to try and understand then what was going wrong in the cancers. So some of the details don't matter here, but we used normal healthy tissue and we prioritized genes in our classification approach that we knew were important in the bladder biology. To do this, we used a network approach. Now, when you create a network from genes, you create a correlation matrix. You say, how well correlated are the expression values of one gene compared to all the rest of the genes? You link them all together, and then you do this approach called selective edge pruning to keep the ones which are really, really important. And in our approach, we then prioritize particular genes which we thought were really important. This allowed us to select almost 100 transcription factors which were really, really well connected. Now, this is a volcano plot on the right-hand side. Now, it's quite a complicated picture, but the key thing to take away is that the genes which are in red type, they're associated with sort of luminal biology, so luminal tumors, and the blue ones are associated with basal biology. Now, most classification systems would only have red genes and blue genes. Ours is different in that half of our genes are not associated with differentiation status. That means we're not just going to get that luminal basal split that all the other classification methods use. The key thing with using these different genes, so we found 100 genes that we were able to use to subtype the tumors. And that allowed us to find a novel group of tumors, these novel basal squamous tumors, and their survival was very, very poor, so significantly worse than all the other groups that we found. And when we look at the biology of these tumors, they're associated with a protein called NRF2. Now, NRF2, as the cartoon kind of shows, is a really important transcription factor that cancers can activate because it helps to protect tumors from chemotherapy, from drugs, and from oxidative stress. So everything that the cells can do to try and kill the cancer, NRF2 effectively stands there with an umbrella and protects those cells. Now, this is really, really important, and it's been found as a druggable target in other cancers. But because we found it using expression data, we've actually been able to go beyond the mutation-only approaches used in other cancers. We found that the mutations that are commonly found within this gene, only some of them respond to therapy. So our classification approach found all of these mutations, and it ignored these ones on the right-hand side. And actually, if you look in lung cancer trial data, the mutations on the left-hand side, they respond to a particular treatment, so their tumor size goes down, whereas the other mutations, there's no effect. So that means that our work here with bladder cancer will also have other relevance in lung cancer. Now, this is just for one target. We're only talking about 5% of bladder cancer cases. Now, whilst in the UK that would probably mean about 1,000 people a year, it's still a small number compared to the almost 10,000 diagnoses of bladder cancer that we get, of muscle-invasive bladder cancer that we get in the UK each year. So what are we going to be able to do about these people? Can we classify everyone in one go? Well, I think not. The classification approaches that have been used so far to try and get all the tumors classified in one go haven't worked. So what we need to do is to ask these biological questions. So take the biology that we understand about the tissue and use that information to try and create subgroups that match to the clinic. The idea here is that rather than trying to do it all at once, we can have hypothesis-driven questions that help us to find groups that we can take forward to the clinic. So that's the challenge. That's what my lab is trying to work on. And hopefully it's something that your project will be able to help us do as well. So thank you for listening. Looking forward to talking to you more across our sessions.